I did not include basic.
We are live.
Okay.
Okay. that's okay. All right. Okay. So thank you so much, everybody, for joining our event. Thank you so much, Dr. Chandana, for taking time out on this auspicious day of Republic Day on 26th of January at 2 p.m. And it's really great that you are here with us. So, not wasting much time due to time constraints as well, I would like to request Bhakti Sadguru to please introduce Dr. Chandana Basu today for the event.
Thank you so much. Hello everyone. Let me quickly introduce you all to our today's speaker. Dr. Chandana Basu is currently working in Center for Genetic Disorders PHU. She did her PhD in molecular biology from University of Tartu, Estonia. Thereafter, worked at the Rosalind Institute, University of Edinburgh, UK as Marie Curie Fellow. After spending almost a decade abroad, she joined PHU where she started her journey as an independent researcher. Her keen interest is in understanding the basis of human diversity and harnessing this knowledge for the betterment of healthcare. She had she has worked on skin pigmentation genetics, lactose intolerance, etc. She currently investigates why people have diverse hair shapes and its genetics. She has received several awards including Mathrubhumi Ke Gaurav Award, SMBE Young Investigator Award 2019 and was featured as Investigator of the Month in Journal of Investigative Dermatology for her work. She is a great psychom enthusiast, was UK STEM ambassador, and currently co-founder of Genetics for You, which aims at bringing concepts of genetics to school children through comics and storytelling. Today, she is here with us to share her research and career journey. Ma'am, we are very, very much honored to have you today as a speaker. Thank you so much.
Thank you, Bhakti, for the kind introductions. I hope I'm audible.
Yes, ma'am, you are audible.
Yeah.
Great. So, like I would like to ask you one question. Like you did your PhD in Estonia, you did your postdoc abroad, and then you decided to move back to India, and today's Republic Day. How do you feel contributing to the STEM research in India, and also at the same time running genetic for you very successfully? So, like, how do you feel in general?
I think it has been very gratifying experience for me as the independent group leader. I will be almost finishing 5 years, and
Mhm.
it has been rewarding in the sense that I have been able to contribute to teaching. Postgraduate diploma students, PhD students. I have been able to contribute to research. I've been able to contribute to like women in STEM, inspiring them. And one of the women scientists is soon going to join me.
Mhm.
And of course, through Genetics for You, connecting to Generation Next and, bridging the gap between the science and society. So I think all these has been like really fulfilling for me and it has been a very gratifying experience.
Mhm. Mhm. Thank you, ma'am. So let's start the session today. The participants would be awaiting. So, like, the first thing that I would like to ask you is I guess you always loved science, biology more. But when did you realize, like, you want to be a scientist for real? Like, when was that moment that, yes, I would be one?
So when I was growing up, of course, science was my favorite subject, and out of the biology, definitely was one of the favorites. And
Mm-hmm.
it was that subject that where I would go beyond the textbooks, to get like the extra books, gain extra knowledge. So I
Mm-hmm.
think the seeds of curiosity were definitely in, but I
Mm-hmm.
I didn't think that at that point of my life that I would be a scientist. It was only during my master's dissertation, for which I went to CCMB, Hyderabad, and I worked in Dr. Thangaraj sir's lab. And
Mhm.
that's where I had the first lab experience in general, like how research life looks like, and
Mhm.
uh, how working like in the lab, contributing to a project kind of. That time, like we were working on basically the DNA fingerprinting project, and mapping the genetic diversity of humans. So we're basically involved in the human origins. So
Mhm.
at that moment of time, I think I felt like, yes, this is like my dream job, and this is what I would like to do for my entire life. So that was the moment when I realized that, yes, I would like to be a scientist, and this is where I would like to go, and I have no regrets.
That's the most important thing. That's really good. So, since you mentioned that you always you did your master's dissertation at CCMB and you worked on genetics and everything. So, like when you read in the science that Dolly was cloned, and then you worked in that institute, so like one of the very among the leading labs in population genetics. So like how was your experience there, and what has shaped you along the journey?
Yeah, so my experience abroad, almost like 10 years, involved my PhD in University of Tartu, Estonia, and of course then moving on for my postdoc to University of Edinburgh. I think these kind of experiences abroad, trainings, really shaped me the scientist I am. So they really are a part of my being as a scientist today. And the mentors, I have been very very lucky with my mentors and supervisors who have, like,
Mhm.
guided me and trained me. In
Mhm.
Estonia, like, especially, we had a very independent atmosphere, and it never felt like there was a pressure of PhD, or in general, as such. We had quite a independent atmosphere of learning. And there I basically kind of got introduced on how to we can collaborate, or how we can initiate collaborations, also like how we can work on multiple institute projects as a teamwork worker, and how we can contribute to them. So and beyond that, then even I moved to UK for after the Marie Curie fellowship. So in
Mhm.
that, I really experienced there, so all there, the part of, like, how we can work on mouse models. So that was like definitely for me, like moving from human genetics to working on mouse, and learning the functional
Mhm.
genetics part was very much of core at the very world class institute. So that was very very important for me as in my training as a scientist. But beyond that, University of Edinburgh had, I knew what was the importance of the professional trainings. And there we have trainings from the Institute of Development, free for the staff, where they have like soft skill trainings, where researchers can register and go and attend those. So that really
Mhm.
helped me evolve as a researcher with those trainings. And so I felt like that played a very very important role in my career development. And all these
Mhm.
experiences, I carry them as goodie bags, my goodie bags, in my life, and try to keep on evolving more.
I see. Yeah. So like whenever somebody is like moving from uh, abroad to India, especially. So you have also written an article, Swadesh for scientists, and you have been proactive about returning back to India. So what led you to do this? Like,
So when I returned back to India, I felt like there is a significant gap of successful stories of people who have
mhm,
returned back to India and have set up their
mhm,
labs. And most of the people who want to return to India often feel very overwhelmed of this fact. And because of not
mhm,
having a connection, in an everyday matter with Indian academia, they feel like,
mm,
there are a lot of like unanswered questions in their going on in their mind. How will we kind of settle
mm,
down? Will they, be able to adapt? Will we be able to adapt to the new environment? So I felt like the example of the, bigger pictures, or the successful people are not there. And that led to, thinking of like having a webinar where we can have like, different people from different stages, like early career, mid
mm,
career, uh,
mm,
those who have returned to India and have successfully
mhm,
contributed to setting up
mm,
of a lab, putting them together. And at that time, Karishma was already, leading this talk to a scientist. So I talked
mhm,
to her, and she
mhm,
was very approaching and, kind of
mhm,
telling that, okay, let's go with the idea. So we had this webinar, and then
mhm,
this article was basically the takeaways from the webinar, so that, people can have a guide
mm,
before they kind of, if they feel overwhelmed, then they
mm,
can go about it.
mhm,
And because there is always a doubt,
mm,
how we will kind of fit. There are always myths around that maybe only Jack is working, and, nothing else is working, merit
mm.
is not working. So people are being overwhelmed by thoughts, and it is, however, not the, right case. So I think like
Mm, mm,
be like a guide to for the people who would kind of come back.
mm,
So I felt like this was
mm,
important at that point of time.
mm. Mm-hmm.
Helpful
Mm-hmm.
for
I
people.
think yes, that is really important because like since past many years, I have everybody is saying also during COVID-19, India tackled the COVID-19 vaccine production and everything so efficiently, and everything made in India was really, really a great achievement for our country, I would say.
Yes.
So like meeting famous scientists like Sir Edwin Southern, you must have been very I would say like it would have been a very incredible time for you. So what was the best thing that you learned from such interactions or big personalities like him?
Yeah, so in Rosalind Institute, one of the other opportunities was we would get to, have different kind of inaugural lectures, which were like, journeys of really scientists who have like kind
Mm,
of been successful, and these kind of really famous scientists coming there.
mm,
And the moment I received the email, I was very, very excited, and I, volunteered
mhm,
to go and meet.
mhm,
So there was also a PhD student, and another like a bunch of researchers. I
mhm,
think like seeing him in person, I was very excited. And I think the lesson I learned with all of the successful scientists are very humble
mm,
and down
mhm,
to earth. And so he really interacted very nicely with us. And then if I when I asked him, what was his, kind of the mantra for success, and then he told
mhm,
everybody told me this is not going to work, but I just went for it. And so I think you know my hard work and not giving up kind of like paid off.
mm,
I think for him, like to summarize it in one line was maybe difficult for the whole of the
mhm,
journey, maybe, but I
mhm,
think it was very, very inspiring for me to meet such a great personality.
mhm. I see, I see. Definitely, ma'am. So, like when you were moving from abroad to India, like of course leaving everything behind that you did for 10 years, then now starting your own lab in India. How was your experience during that time when you were shuttling in between two places?
So I think when you come to India, you have to come with a brave heart, definitely. And there is definitely a kind of difference in the work environment, and
Mhm,
there is no point in comparing, what was there and what is here, rather than concentrating on, what is the current scenario, and working towards it. I think the
mhm,
major challenge what I felt was, I think, like too much things on my plate. I felt overwhelmed
mhm,
with like paperwork and administration, and recruitments, mentoring, teaching, research. So I felt like there is too much on my plate, and I also felt that my time in general is not my own time anymore.
mhm,
There are people dependent on me and my time, and so I have to use my time very effectively and productively. So that was definitely, kind of uh, a challenge. Another challenge was, if of course, that there was a period of COVID when, kind of
Mm,
we were set back with our own
mm,
kind of goals or
mm,
objectives of the project, and that time to keep
mm,
oneself motivated was definitely difficult.
mhm,
But
mhm.
I think, yeah, not giving up and going on with my passion, and worked it out.
Definitely. So, like, now, since you had to set up your own lab in India at Center for Genetic Disorders, you have, like, you got your place. I don't know, I didn't see, but maybe probably an empty room, or maybe filled up with some instruments. And then how did you make that space into your lab? How did you crafted that thing? And like, what were the difficulties that you face, and how did you feel after overcoming them?
Yeah, I think each baby step that you take as a PI, you feel like, rejuvenated, because even the
Mm,
first instrument comes, this is like, oh, one tick done. We So it is not like only my specific lab. We have shared like central facilities,
mm,
and like,
mhm,
shared facilities.
mhm,
But
mhm,
in general, setting up or the entire molecular biology lab, like, which I would
mhm,
like need the instruments at it, each step
mm,
meant like a milestone. And then also growing up with the team. So I was not able
mm,
to have my PhD student like till like 2 years because of the COVID. So then like one team member comes and the other. So it
mm,
kind of like growing as a team was, and evolving as a team was also very, very, an inspiring process. And I think when my whole day goes bad, the
mm,
lab meeting kind of like, rejuvenates me again. And so having, sitting with the team discussing science is something like I
mhm.
really love to do that. So that has been, I think, great.
Yeah, that's true. And then balancing your home at the same time. How could you manage so many things at once?
I think I have been lucky to have a very, very supportive, family,
Mm,
and encouraging family. So, and but I would say when we talk about work-life balance, the burnout is
mhm,
real, and it does
mm,
happen. So, and I also think that science is something where you, in order to have new ideas, you need to be creative.
mm,
So a tired brain cannot generate new ideas. So you definitely need breaks in science. So until and unless, like, I also tell my students, until and unless it's a need of the experiment, you do not need to have, come to the weekend. um,
mm,
but you can take the break. So taking breaks after a certain time is important to kind of,
mm,
come back again with a fresh idea, or even when you're writing a paper, to come back with a fresh outlook. And
mm,
uh,
mm,
uh, you know having a partner who is also a scientist kind of like, kind of makes it easier in a way,
mhm,
because, and my sister-in-laws have been really supportive, that anyways, where when I would like go to conferences, they would come and support me in
mhm,
when my child was younger. So that has been a
mhm,
really, really great support from the family, because they then would witness like how I am growing as a scientist.
mhm,
Yeah.
mm, mm.
But I
Yep,
think,
family.
yeah, I think also it is important to tell the family how they can support you, because
Yeah, that's true.
yes, and giving them up heads, that this is where the help would be needed. Yes.
Mhm. So since you manage everything and try to be best, give your best possible at every role, whatever you play as a mother, as a wife, or maybe as a sister, and also as a PI and a scientist. So what would be the one tip that you learned from your career and you would like to give to everybody today, that when everything goes crazy, what is the one thing that women should do, especially, I would say?
I think I'm still learning time management. But in general, I would say that I focus on winning the week rather than winning the day. So
Mhm.
what
Okay.
happens is, when you first thing, it is important to kind of list down what are your priorities for the day. And when
Mhm.
you do that, sometimes, like as students, as researchers, as PIs, something unexpected
Mhm.
comes off and you kind of derail, and
Mhm.
maybe you won't be able to complete. So at the end of the
Mhm.
day, you might feel frustrated that I listed six things and why I wasn't able to do. However, if you
Mhm.
focus on winning the week, then you have a week targets, and then
Mhm.
you know that okay, if this thing has not been done, then you slowly progress towards winning the week rather than winning the day. So there's also a book by Demir, and that says win the week, and that has been incredible to read it.
Mm, I see. I
Yeah,
see. So that was a really great tip for, I would say, everybody in general, right?
yeah.
To manage everything. And also, like during pandemic time, everybody was really, I mean, the science was growing, but at the same time, there were many labs which had odd timings for working, and it was a really not good time for everybody. So, what lessons did you learn, or what lessons did you think the pandemic gave to the scientific society in, like, if you talk about academia per se, and also like for living in general?
So pandemic wasn't great, but it definitely gave us many good lessons. So for me, myself in general, when we were in a national lockdown, the one thing which I was struggling was I usually need a proper work environment to work.
Mm-hmm.
I should sit in my table in my office work and work. So,
Mm-hmm.
but when you're at home, then it like on or the concept for work from home. Actually built in with the COVID times. So that time, I wasn't able to concentrate on my work because I did not have that my work environment or the line of difference between my home and my
Mm-hmm.
work. So and I was frustrated in the first month, I would say. Thinking like things are not working, I'm not able to finish what I wanted. And then it led to me thinking like if I do not utilize my time, even like shorter or the longer breaks that I have in between the home responsibilities, then it's not going to work for me. So what I did was I then started utilizing the shorter breaks for emails
Mm-hmm.
and other stuff, and then I would lock myself in the room to have a complete work environment for like 2 hours and tell I will be working for 3 hours or whatever. And that's how so I think I adapted to the situation and got a way to work it out during the COVID. What it also allowed me at times is when we are in our career towards this rat race, we know that okay, after PhD there's postdoc, then it's a work, and like this we are just going up the ladder without thinking sometimes, or just focusing on the other goal, one finished another goal. But I think COVID also gave me time to reflect, which we do not do usually. So that gave me that pause to reflect that okay, these are the kind of trainings or where I would need improvement. This is the area where I would like to have more formal trainings or something like this. So I think that gave me that kind of pause to reflect that. And also during COVID, the students were very demotivated. So we then I thought of starting the BHU Women in Science lecture series. So we just needed an online platform, which like
Yeah.
graduate students of BHU, like they're very helpful in providing me that one because they had already one running. So that was and with that I could keep up with the science, and listening to the lectures keep me motivated and inspired. And there I was involved. So we would have like one lecture every month with one national one, and then one international speaker. And then we would involve the BHU faculties as chairs who would introduce them. And so it was like an involvement where, and I think students felt encouraged. And slowly we started getting like 300 registrations from all over India. So it was like one of those baby projects during COVID. Uh, and also I think I contributed to the BHU Serosurveillance Consortium COVID genomic studies during that period, because we thought of like, okay, this kind of can
Mhm.
work for us, and that's how we kind of went upon by the COVID.
Mhm. I think it's really important to, see also good things when something goes wrong, and you really correctly mentioned all those things in a way, because like, somebody told me that don't just count the bad that goes on, also appreciate the good things when they happen with you. So I
Yeah.
think that's a really important lesson that, like, in general, I would also learn from you today.
Yeah, and I would also like to add that there was a lot of misinformation going on.
Yeah.
So then at that time, like, it was also an important responsibility as geneticist to kind of,
Yes.
talk to people, and
Mm-hmm.
reduce this misinformation, and tell them, people at that moment of time, we
Mm-hmm.
knew what our genetic literacy was, and then people knew what mutation was, people knew what PCR.
Yes.
So I think
Yes.
then that was also another role, which was important or
Mm,
contributing towards that.
mm, I see. So, like, also considering those times, and also like today's title of our, event, understanding the genetics of biological traits for public health. So, like, I would like to ask you on this, that how would you explain this to like a layman? I mean, let's say my mom, that they are not from science, but I'm doing my PhD. But so, like, for you, if you are like a scientist and you go out, and okay, let's say there are people from villages, or who don't understand science that much in detail, how would you make them understand the title of the event today?
So basically, I study biological traits. So we all have know that human about the greatness of human diversity. So we have
Mm,
different traits. We can have see people. So when you walk into a classroom, or when maybe you walk onto a
mm,
marketplace, you can see people with different eye color. You can see people
mm,
with
mm,
different skin color, maybe different
mm.
height, maybe different
Yeah.
other features that you can point it out. So we need to understand these traits. Why? Because until and unless we understand the genes that are actually, behind the curtains, or cause, of this trait, then if something goes wrong, which we called as a mutation or a change in the genetic code, then
Mm,
until and unless we know the genes, we cannot, until and unless we understand the normal, we cannot understand the abnormal.
mm,
And in the same
mm,
case, so we have to first go with understanding the trait, then only we can say that, okay, there is a change here, and this leads to
mm,
this disease. So it is like
mm,
forms a crux or the basis of anything.
mm,
And as a scientist, or having, developed this as a niche of my own, what is interesting for me is that when you connect the stories, so you have, like, we work with the insights from archaeology, from
mm,
linguistics, from anthropology, and when we, kind of put these together to understand the human evolutionary trait, because all of us
mm,
can see the diversity, but we do not know what led to this diversity. So it
mm,
may be an adaptation which, would have helped the ancestors, and that's why they got selected, and that's why we have today that trait. So so it is very intriguing to kind of, go back to the human evolution and understand the landscape. And sometimes even after 10 years of research, we are not able to understand the story completely. So it requires definitely, like, people from, like, different disciplines to kind of work on it. But I think putting this puzzle together uh, is very interesting.
I see. That was a really beautiful statement that you made, and I really loved it. I think there should be like a quote for the summaries of today's event, that understanding normal leads us to knowing, what is uncommon.
Yes.
Something like that. It
Yes.
was really beautiful, I would say.
And I would
So,
say in the grant applications as well, I
mhm,
tell, or wherever in the interviews, I do tell that it is a basic science, that what I'm doing,
mm,
because I
mm,
know that, there is importance of translational research as well. But
mm,
this sort of basic science has to be done in order to have, further inputs in the translational research. So I do
Yeah.
say this is a basic science, what I'm trying to do, and maybe its application would come definitely. But
Yeah.
unless we do this, we cannot go for the applications.
That's true. That's true. So, like, since you mentioned about the application and the work taking it translationally ahead, like, what do you think about the today's population genetics and also your work? How can you connect it with public health per se?
So, for example, what I'm studying now is genetics of hair shape, and for many
Mm-hmm.
people, it might be a little different topic in general. So, I'm trying to understand the genes that kind of make our hair straight
Mm-hmm.
or curly. Now, what is important in these kind of hair variants or which are controlling the hair shape is they are also positively selected in so they stand in the like the top list of the positively selected genes. Now by the term positively selected, I mean that they have been favorable for some sort of fitness, or and that's why they have been increased in the number in the population, those variants. So so sometimes what happens is what you can see, maybe the change is the hair shape, but there might be a hidden another trait linked to it which can lead to fitness in general. And therefore, by exploring this, we may be able to go into deeper into that trait, and then decipher why this kind of what is the significance or evolutionary significance of having a diversity in hair shape. If we kind of see this objectively, then we will be able to answer those questions, those hidden questions, and that's how we understanding these complex traits creates a dearth of information which can then be utilized for understanding the others.
I see. And like adding up to this question, what led you to study this, fantastic topic of genetics and of hair shape?
So basically, during my PhD, when I was studying genetics of skin pigmentation, at that time I found that this is quite an not studied topic much, because even after 10 years or 15 years of GWAS, we had at that point of time only 17 loci known for contributing to hair shape at that point of time. So I felt like it is not very well studied, and after my PhD, I thought I'll give myself this is something so I was very keen that I don't want to randomly apply for postdoc. I would like to follow on a project that would I would really like to work on. I was lucky to be employed by the university post PhD, so I did not have to worry or have a tension of having a job later. So that way I was lucky, and so I felt like this is quite unexplored topic. So I thought like I really would like to work on this, and first of all complex traits, and that also specifically hair shape. So definitely it was very difficult to kind of have sponsor institutes or have people who can work with you. So at that time when I was looking for institutes who would like be my sponsor institute for the Marie Curie fellowship application, again having this narrow area, I like was very very difficult to find somebody, but eventually it worked, and I was happy that I could explore.
That's that that's really nice, I would say. So like since you also mentioned, I mean, I have read few of your articles and you have also worked on, lactose intolerance. Like what is lactose intolerance at the first place?
So, milk, we all know it as a balanced diet, and we think
Yeah.
like, we all, it is like the superfood, and, we can all have milk.
Mm,
But
mm,
but what happens is we have an enzyme called lactase. So
mhm,
which is in the newborn, but once the child gets transferred to the solid food, then
mm,
that our production of lactase kind of weans off, and
Mhm.
that's why but and therefore, like, we are not able to, like, digest lactose to the extent as we
Mm.
would have been as children. So but for some people, this enzyme continues to be produced even in the adulthood. And
Mm.
so those people are lactose tolerant individual, and those who had to shut down the lactase gene, and or
Mhm.
it has been weaned off, or and then does not work, are known as lactose intolerant. But that
Mm.
doesn't mean that we cannot drink even a glass of milk. By term lactose intolerance, we means that if you are given, like, 300 mils of like raw milk, and you kind of have it, and if it causes like issues like bloating and indigestion, which
Mm.
means like you are lactose intolerant.
Mm.
However, if you can kind of digest it, then it's called as lactose tolerant. So
Mm.
in my study, which we did, we were trying to explore the SNP or the variant that. So we thinking, like, we all of us usually have milk. We thought, like, we'll have a majority of the population who are actually lactose tolerant, maybe. But to
Mm.
our surprise, we saw that first of all, we had very less, like most of the people cannot actually digest milk in India.
Mm.
And then although we did not take the phenotype scores, this was more on the computers for simulation. But then it also brought about a very interesting story. So the populations where the frequency was higher were the pastoral populations, or those who have had a pastoral history. Now this made sense because and they can really have like 5 glasses of milk and still be okay.
Yeah.
For
Mm. Mm.
example, we have the Roar population in Haryana, who
Mm.
usually take, like, have milk in their culture as such.
Yes.
And there in that we had a higher frequency. And also Toda of Nilgiri Hills, there's also another
Mhm.
pastoral population where the variants. And another result that we got was that the mutation,
Mhm.
which was omnipresent in European, we had the same mutation in Indian population.
Mm. Mm. So it's also genetic. So I have heard that it's also epigenetic, but today I also heard that it's genetic as well, not
It,
just
it is
epigenetically modified.
it is genetic, yes.
That was really interesting to learn something new. So, like as a STEM ambassador, you have worked with school kids as well. So, what is the most fun part about sharing science with them? Because they're so naive, and they get things so very quickly. And like, what is your takeaway from this entire uh, community that you are running?
So I think, like, as a scientist, I feel responsible of communicating science not only to
Mm-hmm,
the scientific audience through
mm-hmm,
publications or in the journals, but also to the
mm-hmm,
non-scientific audience. Because largely, if whatever we are doing in labs doesn't,
mm-hmm,
go to the society, or they are not, they don't know what you're doing, then it's
mm-hmm,
kind of half done. So so I always feel that there is a need of taking your science outside the walls of the lab. And that got its wing when I was working in UK, and there the science communication is really at its up level. And I got trainings, like, how to communicate science in a nutshell, or what are the things that you should use, like you should use layman terms and not the specific jargon
mm-hmm,
when you're communicating science. um, but and that kind of up leveled my enthusiasm for science communication. And back when I went had my own group, I thought, when I was settled in the first 2 years, then I thought, I'll take it up. But I think what really drives me there is their innocent questions, and the light on their face that you can see when they have understood the concept. And they also question you, so that also becomes a part of your learning.
mm-hmm,
And if you have
mm-hmm,
to explain something, at really to a class 4 student or a 3 student, class 3 student, then you have to understand the basics very well. So you kind of also evolve as a researcher because you try to use metaphors, you try to use simple language, so you kind of grow in that. So I think it's always a win-win for a researcher if one does also science communication.
That's certainly true. That's certainly true, I would say. So would you like to also shed some light on your initiative, genetics for you, and also like what do you think that how is it shaping and helping India in longer terms?
So, Genetics for You, for others those who don't know, is a project which has been funded by India Bioscience, and we got this through the third India Bioscience Outreach Grant, which is focused on the early career researchers. So it kind of gave us this opportunity to do science communication, and through Genetics for You, our aim is to basically, because genetics is always termed as a very tough nut to crack. People think it's like very complex subject. So
Yeah.
our idea was to bring the concepts of genetics to
Mm-hmm.
grade 9 to 12 students. That's where we kind of target was our
Learn.
target audience, and to explain them in a very fun and engaging way, involving
Mm.
illustrations, storytelling, and comics. So and our initially our the first year was basically doing the events in the schools around Varanasi, and then in the second year we got extended to the other areas. But our whole sole aim is to put it on a pan India level, and and so that like not it is not only us, but the teachers, the biology teachers can use and these resources and then take this to
Mm.
their classrooms. And it was also important for the students to see how the experiment was done. So through this events, we also do a science activity or an experiment. We call it, like joy. So Joe basically, and the joy with experimentation,
Mm.
and we have a mascot for it. So so that when you see students knowing or studying about DNA, but when they actually see the DNA, they feel excited. And I think getting to do those experiments in hand, we that is like really was crucial, and then students really got overjoyed. What has been so we are three team members in that: me, Dr. Garima, and Nandini. And
Mhm.
Nandini is a part of like the comic library, and so they were very instrumental in turning these facts or stories to
Into video.
and her team in the illustrations or comics. And these comics we are like already have some resources, free resources in the website. And we have also had volunteers who have kind of done the regional language translation. So besides English and
Mhm.
Hindi, we also have in Marathi, in Tamil, and Telugu. So it's like growing. And with
That's
our
really nice.
yeah, and with our like last year we had also like a more scientists contributing to this
Mhm.
project. So we then paved our way to understanding concepts like microbiome, human
Yeah.
evolution, and health and nutrition. And we thought like with comics the visuals stay, and so the concept
Mm.
sticks with the kids. And we have had a very good feedback from the students, and they came and tell us how excitedly they liked it. They
Mm.
were happy. For example, we used this play doh for understanding the animal cell and structures, so inside
Mm.
the cell. So once then they were making, it was so creative at how they would like because each of the group was making in their own style.
Mm.
So they were also using
Mm.
creativity in it, but the teacher said now they will never forget what is inside the cell. So so by
Mm.
thinking that, they were also involved in doing that. And I think like that was very important. Total we have impacted over 8,000 individuals uh, in 2 years, which has been a great journey. And we would like to go on and have more people on board, and as a collaborative effort to take it to their respective, cities, and elsewhere.
I think that is really nice initiative and real I would say motivating initiative in a way that school kids are also learning something. I remember when I was a school kid and there was a big biology laboratory in my school where we were never allowed to go during ninth and 10th standard. And then I took up biology, and I remember I saw the DNA, or the isolated DNA, for the first time when I was in my university. So
Yes.
it took me a really long, to keep on this topic and pursue it as
Yeah.
a dream, that okay, I want to. And like, if I would have seen the DNA when let's say I was in ninth or 10th standard, and then if there was somebody who can explain me in really simple way, then maybe I would have understand the concepts and the topics even better at
Yeah,
that time.
I think like these comics are really engaging, and they are in the form of stories, so it helps to understand the
Mm-hmm.
concepts, and students can very much relate to it. So, and I think there was also a suggestion for us, like, instead of genetics for you, it should be science for you, because there were
Yes,
more, people from physics and other fields
other
who
fields
wanted
as
to
well.
join hands
Mm,
with us. Yeah.
mm,
So I think it has been very motivating, and
mm,
at the end of each science communication event or a school event that we have, it brings a
mhm.
contentment, and
Yeah,
your heart is full. And this is beyond any publications or awards.
definitely, definitely. Let's say a child and coming up and saying, "Hey, Dr. Chandana, I know you. You have been
Yeah.
here," and
I think
I
the
think that's
major
really.
impact was from the participation in India Science Festival,
Mhm,
where
mhm.
we were chosen to kind of contribute through a workshop, and there, like, you could see the real live experience of how
Experience.
the public is kind of understanding your science.
Crazy.
So we had also like a magic calendar, which we initiated as a part of the Advent calendar we have during Christmas
Mm-hmm.
holidays, where we had fun facts released every day. So we put up an exhibition of those fun
Mm-hmm.
facts, and they really went through that, and they were like very interested, and it was like an involvement of everybody there, uh, and I think the most one of the most memorable memories was when you know uh, a grandparent who was a biology teacher was explaining that concept to the
Mm.
grandson, and then everybody in the family did that.
That is something you see on spot, that okay, this is what is translational
Yeah.
science, or the translation work that you are doing. That's really motivating, I would say. Yeah, certainly. So I'm sure, and also throughout your journey, you want to help so many women to get into science. So I'm sure there are so many women today who are listening to this events, and we get this, questions every time, that please, like, we do also something in context of women in STEM. So since you are here today, and you are quite motivating, and also women in science, like, what tips or what suggestions you would like to give, especially to the women or the girls who would have graduated and looking after the career goals ahead?
Yeah, so one of the suggestions would be like to setting up your boundaries. So
Hmm.
there is a leaky pipeline, we know, and a lot of girl students after marriage maybe not are not able to continue or further drop out when they have a child. So I think
Mm.
it's important to set up boundaries way ahead to know what is compromisable for you and what is not compromisable for you, and putting that ahead in the time frame, and this will definitely help. And also I said, you may have a family very very supportive of your career, but you need to train them, and this is also told by Dr. Smita Jain from Cactus Communications during one of her YIM meetings. And then she told that it is very important to train, like, how the family can support you. So
Mm.
having, like, for example, a house help, but then you have to train how they can support you, and then acknowledging them
Mm.
will, like, work wonders. Also, basically having a women in science support group in your
Mm.
workplace, like a small group where you are able to discuss your thoughts freely without having any judgments, and with the trust that this will not go beyond this, this small group, and taking each other's viewpoints, and this kind of really helps. also finding mentors beyond your own institute or your workplace definitely works, because I have been very, very grateful to have such mentors which whom I met during the YIM or the regional young investigator meetings which I organized. They really would like to pull you. And so these kind of and last of all is to not to stop dreaming, to dream big, because until and unless if you give up on your dreams, everybody else would. So you have to keep up your dreams high, and then only you will have wings to fly.
That was really motivating. I'm sure that many participants today, especially girls and women who want to pursue their career ahead in science, would reach you out and also like
Yeah,
talk
yeah.
to you once
I'm more
again.
than happy and approachable through email, social media, whatever.
Yeah, yeah, absolutely. So, like, I would like to ask you two last question of the session. One question I would ask now, and one question I would like to ask you after the presentation, like a closing
Okay.
one. So, like, what tips would you like to give today to, like, I would say bachelor's or master's student, who are looking to pursue their career ahead, and also like people like us who are pursuing PhD right now, like back in your times, like what would
Yeah.
you like to?
So I think in during my in these last 5 years when I returned to BHU, I had a lot of students coming up, very, very confused about their careers.
Hmm,
And
hmm,
so what happens is that sometimes students feel a lot of peer pressure, that
hmm,
these people are doing, maybe I'll also go their way. So I
hmm,
will tell, and sometimes they are, more skeptical about the scope of the subject in general.
hmm,
I'm
hmm,
taking this field now, what is the scope of the subject in future. I will
hmm.
tell that you don't have to rely on the scope, you can create the
So
scope.
create
So
a scope.
don't follow the trend, be the trend. So
Hmm,
I would say that if you have a passion, for example, in following forensic science,
hmm,
uh, then you just follow it because you have passion. And if you make a compromise with the peer pressure that and move on to a
hmm,
different subject, it will make
hmm,
like life not more cumbersome for you, right? So you
hmm,
should follow your passion, whatever you really feel or enjoy doing that. You should make that. For example, I had a student, I would not like to mention her name, but she came to up telling that she was really wanted. She has done masters in forensic science, and
hmm,
she really wanted to do a PhD. But a lot of her friends were
hmm,
telling that the scope, this field has no scope, and
hmm, hmm,
we are moving to other fields or whatever. So she was very confused. But when she came to me, then I told, like, you can, if you have that then you can work towards it. And then I
hmm.
get an email after 1 year telling that I have got the prime minister fellowship, and in PhD, doing for pursuing PhD in forensic science in
Fantastic.
IIT. And the first, after taking admission, you're the first person I'm emailing. So that was
Ah,
very, kind of touching for me, and that made my day. So I think you should be the trend rather than follow the trend, I would say. For PhD
hmm,
students, sometimes I know that in the first years, you're not able to maybe create your niche or find your niche.
hmm,
And that happens, and in that way, like, the rotations or lab rotations works or helps. But in universities where you do not have lab rotations as a part of PhD program, you can, I think, getting to know well beforehand about the group and the work the group does kind of will be the key for those students. And so that you just don't accept the project as such, but see if it is you're genuinely interested in that project.
hmm,
So
hmm,
this is very, very important to do your homework about that before you join a team or
hmm,
join a project. So I think
hmm.
that is really, really helpful in that sense. And I think
Certainly.
now with the UGC new regulations, there is no the subject bar, which
Yeah.
kind of resonates, that people don't have to worry about that, I have this,
Mm.
or what is, where I should only use this subject because this subject has more vacancies. No, you can uh, switch on subjects, and this has been transformative. I would say a great initiative.
That's really nice. So I also remember, my story. I also got an admission in forensic science, but I continued with molecular biology and like yeah. But it's really interesting. I really it was really motivating to listen today about like pursue what you want, like nobody usually comes and suggest in a way when we ask for help that okay, I want to do this, and like there are really few people who come and tell, okay, you want to do this, go ahead, don't
Yeah.
worry about it.
And you should not hesitate to ask help. As I said, you should
Yeah,
even meet outside your own department. You should go and
that's true.
approach, because until you take actions, actions will not happen, and successful people take actions. So you have to that
True.
person may say I don't have time or whatever, but until and unless you ask,
Mm,
you will not get that help. So I will say that,
mm,
oh, if you really are inspired by somebody, or you would like to talk to somebody, go and talk to,
yeah.
approach that person, and connect, and networking. And in nowadays, like with the internet, like the worst thing you can do is not being aware.
No.
So you have everything on there. So I think you
Yeah.
should work towards the goal, and have a timeline ahead, that this is my timeline. You should not work aimlessly. You should have like a 5-year plan, or the next 2 years plan, that this is how
Mm.
we are going to move forward.
Mm. I think that was really motivating for me personally as well today, to talk with you in context of this podcast. Like it makes me more stronger to even work more for the psychom synapse as well and
Yeah.
continue the STEM journey.
Yes, yes, definitely. You people are doing a great job.
Thank you so much, ma'am. So we can have your presentation now,
Yes.
and so you can
Shall
share
I
your
share?
screen.
Yeah.
Yes.
So, is it visible,
Um, yes, ma'am. It's visible.
and is it full screen?
Yes, it's full screen.
Okay. So, shall I go ahead? So, can you see moving of the slides also?
Yes, ma'am.
Okay, great. So, today I'm going to make a walkthrough about understanding the biological traits and their importance, given an introduction of what my lab does. And in terms of research, although there were many projects, I just chose, due to constraints of time, two projects that I will be speaking about. One is the genetics of skin pigmentation, very close to my heart, and the another one is genetics of fingerprint patterning. And I would also like you to share few glimpses of my psychom journey of genetics for you. So, understanding basically the genetic basis of phenotypic diversity. So, to understand the term genotype and phenotype. So, genotype is basically the genetic material we have, is called as genotype, and phenotype is what we see as the external characteristic of that individual. Now, a lot of time, as I said, we happens that the diversity we see, we think like it is just there lying as such, but usually so we had in our human evolution basically two major time epochs. That was one where the ancestors moved from the African homeland and they adapted to different environments. There were also adaptations to diet and lifestyle. And then, so understanding this in the broader context or in the bigger questions tells us like why there was a need of this particular diversity, of this particular adaptive trait, what has been its evolutionary history, and what selective pressures have shaped the current variation that we see today. So, as I said, unless we understand normal, we cannot understand what's abnormal in case of disease. And therefore, using these, understanding these variations that are a part of natural variations of the natural diversity that we see, we can harness information for the public health. So our lab, which is known as GenoPhen, that is genetics of phenotypic and adaptive traits, is what we are particularly interested in studying. We are working with a PhD student on skin pigmentation. Then we are trying to understand the genetics of hair shape, lactose intolerance. We are also working on Ladakh populations in some collaborations with the Ladakh Institute. And then we also worked on understanding of genetics of fingerprint patterning. So when we talk of these complex traits or skin pigmentation in general, skin pigmentation is not being controlled by one gene, but it is many genes. And therefore, understanding the skin pigmentation is not straightforward, or genetics of skin pigmentation is not straightforward, but it's very, very complex. And the biological component that makes our skin lighter or darker, or what is contributing to our skin color, is called as melanin. Now if we say call of this, for we can compare this as a train having different bogies, so where each of these bogies is contributing to the journey, right? And in the same way, we need to know the term which is called as effect size. So contribution of each of this gene towards that trait, or the variation, or the effect, is called as effect size. So if we think of basically an orchestra, or a people of people playing together in symphony, then we might sometimes hear the cello, for example, in a louder voice, right, than the other. Or maybe if in terms of Indian concert, maybe the flute is having very high pitch. In that case, so these are the genes which are having higher effect size, or contributing more to the variation in compared to the other genes. So this is like a measure of how much a gene is contributing. So that is called as effect size. Now, when we have many different genes which contribute to skin pigmentation, but there was a discovery in 2005 which was published in Science, which told that there is a gene called as SLC24A5, which is a pigmentation gene, and they were doing an experimentation on zebrafish, and basically a change in the third exon of this gene led to basically lighter zebrafish, or a paler zebrafish. Now, when they kind of reciprocated the study with the humans, because since zebrafish was just a model organism of studying, then they found that yes, this variant has a very high effect size, and it explains about 25 to 38% of the differences between the skin color of the European and African populations. And in that particular study was done on admixed population. So, and talking like how our skin pigmentation has been shaped. So it has been shaped by the natural selection in response to the UV rays. And so this light skin mutation in this gene was helpful, or being helpful in to the population, and therefore was positively selected because it was contributing to a higher fitness. And so now we have in Europe like almost 98, it's almost like fixed, we have 98.7 or 100% of the population who has this mutation, whereas the ancestral one is present in the African and the East Asian populations. So our question with our study was, we wanted to know what is the phenotypic range of skin color in South Asia, since no other there were studies on Indians based in UK or US, but there was no study done till then like on the ethnic Indian populations. So we wanted to know what is the phenotypic range. And if you see, or importance of seeing India in the context of a study of genetics of skin pigmentation, then it kind of has a mosaic of skin tones. We can have we do have yellowish skin tones, we have pinkish skin tones, and then we have brownish skin tones. So we have a range to explore, right? So we were interested to know the phenotypic range, as I said, and we were also interested to know what is the role of this particular candidate gene in South Asian skin color variation. So what we did was we collected saliva as well as blood, and then we extracted the DNA. So here you can see basically three individuals, and whose melanin index is being given below. So melanin index is the quantification of melanin, and it is when usually measured through a derma spectrometer. Here is the photograph of derma spectrometer. You can see it has two boxes, which is erythema, and which is measures the redness of the skin, and melanin, which measures the melanin content of the skin. And if you have higher melanin indices, it is usually varies between 0 to 100, and higher means darker skin, and lower means the lighter skin. And we, for the genetic purposes, we take the readings from the upper inner arm, which is the unexposed area, because our exposed area will be more prone to tanning, and therefore we can have seasonal variations in the measurements and etc. So we want to go to the there are two types of skin pigmentation, the constitutive and the facultative. So constitutive is the skin pigmentation we are born with, and facultative is the temporary one, which we kind of get through tanning. So in the winter months, for example, we have be lighter. In the summer we get tanned. So when we done the phenotypic distribution in India, we saw that we had, a huge, as we would expect, we had a huge variation in the melanin indices. It ranged from about 28 to 79. And as because also, Indian populations are very diverse, so we had to, have a homogeneous cohort to, for the notification, for noting these melanin indices. The observed range was definitely greater than the Europeans or the East Asians. And then we were interested to know what about the association of this particular SNP that we just talked about with skin color. So we, had, since we had measured like 1 to 8 individuals, determining their range, and then we took the top and the below 10%, representing that very this population, and then we genotyped those. And here, as you can see, we have AA, AG, and GG genotypes, which are the three genotypes of that particular variant. And you can see that AA contributes basically to lower melanin indices, and GG or AG contributes to the higher melanin indices. And when we did the association test, then we found that yes, there was a significant effect of these genotypes on skin color. And the effect size, as I discussed before, as par with the Indian population or the cohort we studied, was about 27%, which was very, very high. We then wanted to know about the evolution, or how did light skin evolve, and what about this mutation? Does it have some kind of do we have a common ancestor of it? So we resequenced this gene in about 95 individuals, where we had representative samples from CEPH of Africa, North Africa, Europe, South Asia, North and Central Asia, and then we resequenced the entire gene. And then came up this question that this particular light skin allele, which is common for Europeans and South Asians, is it identical by descent, or do they have a common ancestor? And our haplotype or phylogenetic analysis based on this resequencing data that we did showed that indeed, yes, they had they shared the same mutation. There might be other mutations as well, but as far as this mutation was concerned, yes, this mutation was the same as that of the South Asians and Europeans. This was a part of the photo shoot, which was done for the press release of my study, where we have Indian and a European child, and what connects is the golden gene. So that's how we portrayed. So, and my next topic is about genetics of fingerprint patterning. So we all know that Each of our fingerprint is unique, but there are common patterns in our fingerprints, which we, if you would see closely, are basically three major types. We have loops, we have whorls, and we have arches. Now, when we kind of subdivide into again six types, then we can have as simple arc, tented arc, and a loop based on which side it is. We can have ulnar or radial, and we can have simple whorl or double whorl. So, so when there are what you see now in the picture are basically minutiae. These are the features in the fingerprint. It can be, for example, a double bifurcation. It can be a eye. It can be a bridge. It can be a crossover. Now, when two persons of the fingerprint matching is being done, as you can see in for forensic purposes, maybe in serials or movies or in real life as well. When this matching is done, then what they are matching actually is these minutiae. And each country has the minimum number of minutiae, like an index, like 20 or 12. Like each country has its own index, which should be matched to tell that yes, these two fingerprints are of the same person. So that is how the fingerprint matching is being done. So we were interested to know the genes behind this fingerprint patterning. What exactly, which are these genes which kind of lead to fingerprint formation. And for this we conducted a GBOS. It was together a part of a collaborative work of a very international team, and where we kind of studied the fingerprint patterns of all 10 digits in 9,000 individuals in 3 Han Chinese cohorts. And then we identified basically the 18 loci which were contributing to the fingerprint patterning. Out of this, the topmost signal was the EVI1 gene, and this was actually known in embryonic limb development. Now, this was quite surprising for us because usually when we talk about fingerprints, we would expect the skin, the skin genes or any of the skin formation genes, coming up. But then what came up was this embryonic limb development. So we thought of doing the functional experiments using mouse models to see if EVI1, whatever hit we have got, is the real one or not. So when we tested the mouse models, we found that basically the genetically modified mice had with decreased expression of EVI1 had developed abnormal skin patterns on their digits. And because of that, we thought that maybe these digits can be a homologue for the fingerprint in mouse. Now, the mouse does not have a fingerprint on its own, right? So these digits that you can see here, where basically we kind of each, so here each of these fingers are called as digits, and there we have a certain markings. So I had a very interesting time exploring the method of devising a method of scoring these patterns. So we developed a coding for basically, so we if there was a continuous from one end to another, we would score it as continuous. If there was dashes or broken parts, then we would score it as discontinuous. If it was in only broken in 2 parts, we would score it as incomplete. And then we scored the mutant and the wild type of the EVI1. The heterozygous derived ones were actually lethal, so we had only the heterozygous and the wild type to be compared. Homozygous derived were lethal. So we found, when we compared, then we found, and we did all those scoring, and once the score was complete, then we found that the reduced frequency of the continuous ridges, with digits 3 and 4 having more discontinuous ridges. And this showed that yes, the effect was also evident of EVI1 was also evident in the mouse models, telling that yes, what we have seen as a top snip or a top signal is in fact validated through the mouse model. So then we did the meta-analysis, involving other studies, and then we found that which now the individuals were turned up to about 23,000, and then we identified about 43 loci, which mapped to 105 notable genes, and these again the effect size coming to 4.6 to 7.9%, explaining the fingerprint patterns. So we also wanted to know if there is an association between the fingerprint patterns and the hand types. So we found that there was a correlation between the fingerprint patterns and the limb-related hand phenotypes. So we measured the hand length and the digit length. So we found that the people who have more whorls had longer little fingers. So this kind of was an association that was seen. This was published in Cell, and to summarize, we actually found 43 fingerprint associated loci, and the interesting result was limb development genes underlie the variation in fingerprint patterns. And if we then, you know, uh, um, go back to the gestation period where actually fingerprints are formed, then they kind of coincide with the same gestation period when the limb is formed. So it kind of makes sense. We also, noted through our study that EVI1 is the top signal, and it alters the dermatoglyph or the fingerprint patterning in the mice by modulating the limb rather than the skin development. And fingerprint patterns are genetically correlated with the hand and finger proportions. Speaking of the SciComm journey, so basically taking my research outside the walls of the lab. So as a PhD student, my first SciComm experience was when, there was Estonian radio who wanted to, discuss our research published during my PhD, and they came for an interview. And after that, we did this photo shoot, and so it kind of that was the beginning of my SciComm journey. Later, when I went to UK for as Marie Curie under the Marie Curie Individual Fellowship, then I got more opportunities of training, and I signed up being a UK STEM ambassador, through which we would get invitations for volunteering for the community events, so we could contact the public in through them in the community-based events. I also contributed to different open days and soapbox science. So in Genetics for You, which we kind of is funded by the third India Bioscience and the fourth India Bioscience Extension Grant and Outreach Grant, respectively, we had three primary goals, basically to teach the concepts of the genetics in a fun learning way, to create resources that can be used by teachers, parents, and students, and to give them the joy of experimentation and what actually experiment looked like. So in this, we had very many events, but it was mainly targeted to the students of 9 to 12, because we wanted, that was also the same time that they would, study about DNA in their textbooks, but so they could have related that, and that our main motto was to make them as responsible citizens when they grow up, so they could have that exposure towards genetic counseling or about the genetic variants or the genetic diseases, and therefore can act as more responsible citizens. So there is a process flow. We have a team of geneticists, currently me and Garima. But then, when we kind of contact other subjects, then we have a subject expert. And then we have Learn with Comics, who are our collaborators, and they create it into, dialogues or in a, really funny illustrations in the story. And then with the help of volunteers, we take it to the schools. So these are our resources that we have created. We have created the Advent calendar, where we have fun facts. We have comics in English and Hindi and in regional languages. We have a DNA in a STEM kit which has been developed and can be requested for, and we have also like fun with crosswords. Here is like an example of the creation, where you can see that um, what is DNA made up of? It is made up of letters representing as A, T, G, and C, and we have 3.3 billion of them. And here, where there is a conversation between a child and a mother, where the child says, see, I'm a left-hander, but it's actually the genetics that make you kind of left-handed or right-handed. Um, this is the glimpses of the India Science Festival 2023, and one of the most memorable event. Most memorable memory was when the cleaning squad, after we had ended the event, they were very closely looking by our stall, and when after the end of the event, they came up and asked us, can we also try? And this then in their own local language, this lady, as you can see, was explaining to others, and then they also performed the experiment and felt empowered. So this explains our motto of bringing science for all or science for everyone. These are again glimpses of the ISF, and where we kind of engaged with people from all ages. We had this exhibition of fun facts, and at the end of the day, it was the smiling faces and basically the joy of understanding a concept. Together, we have like impacted about 8,819 individuals. We have done 10 school events and 4 national events, and this has been really, really motivating. So this is my group, consistently a very small group of about 5 members, and this is our Genetics for You group. And I will be more than happy to take questions. And if you wish to join us, or if you, after listening to this presentation, think that this is something you would like to take up as a research, then please feel free to contact us for internships or PhDs or postdocs. So thank you, everyone.
That was really fantastic today to listen you through the journey and the genetics population. And the most important interesting part for me was the fingerprints and how you developed it in the mouse, and the way you labeled them and everything.
Yeah.
I think it's really, it's something that you can see in while doing biology.
Yes.
Unlike, like, you can see the changes, phenotypic changes, genotypic changes, and everything. I
Yeah, I
think it
mean,
was really.
at that point of time, we were really following science and following curiosity. We did not know
Mm,
where this result would go. So we by
mhm,
scoring developing that method, and after that doing the scoring. So for me, in my area, the most aha moment is when the genotype, because
mhm,
when we do, then the genotype is blinded. So
mhm,
so we have just the individuals and we have done the genotype, but when the phenotype is completely done, so that we are not biased during the phenotyping. So once the phenotype is being completed, for example, noting, so we do not know when we are scoring the digit, whether it is the mutant or it's a wild type.
mhm.
We just do the scoring blindedly, and after that, you do it with just a sample number, and then there is a moment when you put the genotype and the phenotype together, and that is the aha moment when you kind of see, yes, there is association.
That's something really interesting, I would say. So
Yeah.
we have few questions from our participants. So the first thing was that, like, Abida Anand, okay, if I am not wrong, she is, so they are asking that how, or is there any current link for autoimmune diseases and this genetics or something like that,
Oh, so
population genetics.
I think the question might be about the fingerprints, maybe, as I would predict that, because
Mm-hmm,
initially when
mm-hmm,
the research group started, we started as a group in exploring. The whole idea was there are some fingerprint patterns which are being known to be associated with some diseases, but then there's still now we do not have a scientific explanation to it. So we don't know that for sure. So yes, this, like, now that we know the genes, we can then explore whether there is association between the disease and the fingerprint patterning, but for now there is no scientific explanation. But initially that was the that what intrigued of that yes, we should understand this to understand this question. So,
Mm.
so that was the motivating factor of starting the study at the first very point.
Mm. Mm. I see. I see. So I think that was this was something interesting question for me because I never knew about if there was any correlation existing or maybe in past. Okay, we have many questions, but due to time constraints we cannot take all of them, but honestly there are so many women, especially girls, maybe postgraduate or pursuing, who wants to really interact with you. I think we could set up one more session where you can directly interact with students, especially
Yes,
where we can talk more about career opportunities, goals, and some things like women empowerment and things like this. So we would surely come up with such kind of events in future because we have been getting this since long. So we look forward towards this. And so one last question that I usually ask to all the PIs or STEM enthusiasts who I have hosted and also will be hosting in future. So it's like, what is the favorite discovery uh, for you as a scientist, or maybe when you dreamt of becoming a scientist? Like, I would give you a few examples of answers that previous speakers gave. So one of the scientists which I remember, she gave that the discovery of H plus and Cl minus was really important, in the science itself, which also motivated her to go ahead. And then recently we had the director of CCMB, and I mean, there was not something one, but he pointed out many, but out of which like computational and biology, going together was something really good. So what, according to you, is the greatest discovery or the best discovery in the science?
see, each of the discovery for a scientist remains very special to his or
That's
her
true.
core of heart, and because
Yes.
they have put in the efforts. But for me, I think the revolutionary discovery has been the Human Genome Project. So,
Uh-huh.
so the fact that we and if you can see, this was like a 15-year project, but they kind of even
Mm.
finished before the deadline. So the
Mm.
breakthrough that it brought to us,
Mm.
when the human genome code was deciphered, we could see,
Completely.
see it completely. We are still discovering.
Yes.
There are 3.3 billion, but now with when you do the whole genome, and have a higher resolution, you can. So this,
Mm.
I think, you can compare the sequences. You can know where the mutations are. So I think for having a reference genome to start with has been the major discovery for me, and then the most inspiring one. And every time, even when I teach the students about the entire project, how it was done, it kind of gives me goosebumps.
Mm. So again, knowing the control or the normal is important. Then we can make more discoveries for something unconventional.
Yes, I think the population genetics is the crux. For example, in the COVID, also we saw that some people were heavily affected. For example, even at the
Mm.
young age of 23, they had to go to ICU. Some people were
Mm.
entirely asymptomatic, and also we could see, in general, India was affected less than the other populations, right? So what was driving that? It was the ancestry. So also within India, we had some regions which were heavily affected, and some
Mm.
regions which were not. So I think that's a crux for understanding anything beyond. It's like a base, which we know to want to understand, to know. And now with biobanks, and now with the current human genome, with India releasing, just last in within the last week, releasing this, will make much more important discoveries, and will be a source point for more and more discoveries and more translational things, because then we know that if we kind of, the importance of studying different populations is, then if we have a, for example, a vaccine or an antibody, then we know that it
Mm.
will work for all populations if we have kind of that reference to work with. So I think it forms the crux, and it's very important to study that.
Definitely. I think you have motivated everybody today a lot, and also with your initiatives towards science communication and in general, everything. So I really loved hosting you today. It was really, I was a little bit, in a way that, okay, today is Republic Day and what the event would turn up. But I think it was fantastic. I really enjoyed interacting with you.
Yeah, and I just a statement for the viewers or who are online with us today. If you are unable to
Mhm.
ask us the question, you can either send they can either send it to you or me,
Yeah,
and I'll be happy to answer them. Due to constraint of time, maybe I won't be able to answer each and every question, but please feel free
definitely.
to DM me or contact me, and I'll be more than happy to answer the queries or reach out to me. Um, Thank you.
Certainly. Okay, so I would like to ask Somalia to please deliver vote of thanks for all our participants and also Dr. Chandana Basu.
Yeah, sure.
Somalia?
Yes, yes, yes. One second,
Okay.
one second.
It is also possible that he is involved in the live streaming.
Yes, yes.
So,
One second, one second.
This year.
yeah, he's here.
Yeah, hi.
Hello.
So it's my really privilege. Good afternoon, everyone. It's my privilege to extend a heartfelt vote of thanks on behalf of all of us to ma'am. First and foremost, I would like to express my sincere gratitude to our esteemed speaker, Dr. Chandana Basu, for sharing their valuable insights and share insights and experience expertise with us, and your engaging presentation has been both enlightening and thought-provoking for us. It has left us with valuable takeaways and a fresh perspective as well. And I would also like to thank all my teammates for their sincere efforts in putting together this event to achieve this seamlessly, from coordinating the schedule to ensuring a smooth flow, and your dedication behind the scenes has made this webinar really a success. A special thanks goes to our audience. Your active participation, insightful questions, and enthusiasms have truly enriched the session. It is your engagement that drives us the success of events like this. Once again, thank you all for being a part of this wonderful session. Let's continue to think, share, and grow. Have a great day ahead and happy Republic Day. Jai Hind. And please don't forget to like, share, and subscribe our channel, the SciComm Synapse. So we will
Thank
be
you,
posting
everyone.
our feedback link in the YouTube chat box. So it's done. So we have posted the feedback link on our in the YouTube chat box. Please do fill the feedback link form to get your e-certificate. Thank you.
Thank you very much. Thank you. It was absolute pleasure to be with you all today, and especially very gratifying for me to share this experience on Republic Day. So happy Republic Day to all. And I think after returning, it is every time I am in the 26th January or 15th August event, in the unfurling, on your own soil. That feels great. That feeling. So happy Republic Day and Jai Hind from my end as well. Thank you.
Happy Republic Day, ma'am, and Jai Hind to everybody. Okay, so please give us the feedback form, filled. It's really important for us to improve each and every time, also to get new speakers and also to know what audience participants wants to learn from our side. So please fill in the feedback form. Ma'am, I hope you enjoyed today's session.
Yeah, very much. And as I said,
Thank you so much.
open to questions and anything else that I can be of help.
Definitely. Okay, so we can end the live stream. Okay, we can give some time to participants. Mm-hmm, mm-hmm. Okay, okay.
Yeah.
Yes.
Yeah.
Bye,
Thank
ma'am. Thank you so much. Thank
you.
you so much for joining today. Did the live stream end? Seems not it ended.